Hello. All right. Um, to answer your question, yes, this is the bomb lighting that Lizzo talks about. So there we go. All right. I'm going to give you all the secret of fire here. Like Prometheus, see if you can hold on to it, not get burned or chained up and get your liver eaten for 1,000 years or whatever the heck happened. I don't know. Um, so, Negative strand virus polymerases are related to each other, and those related polymerases are similar in structure. And they do similar things. And some of them are real similar, even though they don't seem like they should be at first. So let's get into it. We've got 5 slides in this entire deck to get through. I think you can do it. You can definitely do it. Okay, so we've got to divide up our negative strand virus polymerases into a couple of groups. First group, well, 2 groups. Yeah. The first group are the ones that steal caps. So these are viruses that cannot make their own cap structure. And so what they do is they are going to grab a messenger RNA from the host and decapitate it. Literally, yeah. They don't just take the cap, they take the cap plus something like 5 to 10 nucleotides, depending on which virus we're talking about. All right, how do they do this? And how do they use this for great gain and profit? Well, here you go. So, first of all, let's look at the machinery that does this. And we're going to relate everything to the influenza virus polymerase because instead of being one big long protein, in most negative strand viruses, it's a big long protein, and it's called the L protein, and the L stands for large. Yeah, you heard it here first. The large protein. It is large, to be fair. Usually the largest one in the virus. Yeah. Dumb name for a protein, 100%. But memorable, I guess. So, yeah, the L protein. So, normally there's an L protein, but in this one case, the flu viruses, influenza A, influenza B, influenza C, to Goto virus, things like that, it is broken into 3 chunks. And those 3 chunks align exactly to the 3 parts of the L protein in some of the other viruses. So it's just like a shattered version of a normal polymerase, which is pretty cool, right? Yeah. So our 3 parts here are called PA, PB1, and PB2, and they occur in that order on various L polymerases, so that's a good order to remember them in. So PA, the A and the B stand for acidic and basic. Back when people had no idea what these proteins are, and that was like the one true thing you could actually say about them. Like, I guess it's slightly more acidic, I guess it's slightly more basic. Doesn't have anything material to do with what they are really. It's just, whatever. Yeah, it's a way to categorize things when they had no idea how to categorize these things. Together, these are the polymerase complex. They each do different jobs. So, up front, all the way at the end, is PA, because A comes before B, yeah, and B1 comes before B2. I mean, I know how letters and numbers work, so do you. And so does the virus, for a change. So, PA is going to be the cap snatcher. It is going to grab onto an RNA that has a cap on it, and it's going to chop the cap off. So it has an enzyme called an endonuclease domain at the end, which has a couple of manganese ions in it, and it uses those manganese ions to break a strand of RNA and then keep it for itself. All right. The next member in the chain that we're going to talk about is going to be PB2. So PB2 is like what you would call in a 30s gangster movie the bag man. So PA is actually committing the crime of the cap snatching and then tossing the goods over to PB2. Now, it may well be that PB2 is actually right there, totally willing accomplice, holding on to the cap while PA saws its head off. But, who knows? Yeah, viruses, they're terrible in every possible way. Yeah. So PB2 holds on to the cap. PA cuts the cap and some nucleotides off. It measures it pretty precisely. Then they feed this monstrosity, this sort of head of a messenger RNA, down to PB1. And PB1 is the polymerase. And it's like a normal polymerase. It has a palm, it has some fingers, and it has a thumb. And when they make a complex, it sort of sits like this. There you go. And PA sits on top of the fingers, and PB2 sits on top of the thumb. And so PA and PB2 are right there, kind of at the same spot. That's why I think they're partners in crime, most likely. All right, so what does this mean for me? An arenavirus polymerase, for example. Well, let's go into it. Arenaviruses have one big long protein, or you might say large protein. It's the L protein. At the beginning of that, look what they have. They have an endonuclease. Oh my gosh, and they even matched up the colors. So all this region folds up into the same shape and does the same job as PA. And then look what it's next to. So this one is one big long protein, so it's not 3 bits that assemble in a particular way. It's just physically attached to the fingers region. So in influenza, they make a separate protein from a separate piece of RNA. And it goes and sits right at the place where it probably used to sit. I can only imagine that these things were one protein originally and just got broken up in influenza virus's relatives. And so it goes into the, here's our PB1-like domain with your polymerase core in there. And then you have your PB2-like domain at the end, which is going to sit, remember, on top of the thumb. Boop. So PA over here, PB2-like domain over here, and PB1-like polymerase domain in the middle. And PB2-like domain is going to bind caps, just like always. Yeah, it's amazing. So here you got 2 really different viruses. One has one big polymerase protein. The other has 3 small polymerase proteins. But darn it, if the 3 small proteins don't line up with the 3 parts of the big protein. It's just a shattered version that puts itself back together inside of a cell. All right, pretty cool. Yeah, agree. Fine. So this is actually Bunya virus at the top. Sorry, not arenavirus. This is arenavirus at the bottom. It's got the wrong color scheme, but it's got that same organization. Endonuclease, PA-like region, RNA-dependent RNA polymerase, or PB1-like region, and then a PB2-like region with all the same stuff. Same cap binding domain. They call it domain 2 here, but it's a cap binding domain. And the little lid domain sort of sits on top of it. Yeah, exactly the same shape. So, there you go. All right. Now, what can we do with this? Well, first off, let's see the influenza that we're comparing all these things to. And this is a structure of the big influenza enzyme with the PA, PB1, and PB2, like that. There we go. So here are the 3 proteins shown up at the top. Here is the structure, how they sit. And so you end up with PB1 making up the bottom, and the fingers of PB1 are where the endonuclease sits. So that's in PA. And then back here is the PB2 region, these sort of pinky, reddy, yellowy colors. And those sit on top of the thumb. And it's as easy as that. That's the enzyme. Sort of like a great big cupped hands, kind of doing that, but with kind of a big chunky thing on top of the fingers and a big chunky thing on top of the thumb. Okay, let's look at another one. Here's another one. This is an endonuclease of an arenavirus. Yeah. And arenavirus, look, it's the same thing. See the hand? Well, going like that or going like that. It's like rotated. Here's our endonuclease. So here's our endonuclease here, sitting on top of the fingers. Here's our little fingers. There are these blue things here. You can see the color down there that matches. There's a special thing called a fingertip. Don't worry about the fingertip. Then you've got a palm domain. That's where the catalytic site of the polymerase is. Then you've got a thumb domain, which is this gigantic brown thing. Presumably the polymerase opens and shuts, just like that, just like normal. And then sitting on top of the thumb, you've got this extra region, which has the PB2-like cap binding machinery. There you go. Big difference between these is that is how they measure. The arenavirus polymerase will chop off about 4 nucleotides plus a cap, and after that it starts matching, and that's where the virus starts. Influenza, it's usually 10 or 11 nucleotides. Those are the most common things that it chops off. Yeah. Influenza usually gets its caps from RNAs that it finds in the nucleus. It actually, in sort of a fit of spite, I think, dismantles part of the splicing complex. It rips apart the spliceosome and takes out a thing called the U2 RNA, which is one of those small nuclear RNAs that binds the small nuclear RNA proteins. SNRPs is what people call those. Anyway, and this small nuclear RNA has a cap, and so it gets beheaded, and it just happens to match the sequence of influenza if you go about 10 nucleotides downstream. So all the influenza messenger RNAs have 10 or 11 extra nucleotides of that RNA that they stole on the end. And it's fine. It doesn't matter. Arenaviruses tend to go for 4 or 5 extra, and those are 4 or 5 from one of many RNAs that they also steal. But they also have a sort of a sequence preference. They want the last couple, like after the 4th or 5th, they want to start matching the first few nucleotides of the genome so that they can actually use it like a primer. Yeah, so there you go. All right, let us continue. So here is the big old thing. Since we've got an exam question on basically comparative virology, let's do some comparative virology for you. Why not? Yeah. Okay, so in arenaviruses, they have an extra protein called the nucleoprotein. It has a little cap binding pocket, and so the little cap goes right into there. Great. And when I use this terminology, M7G, M2'ON cap, what that means is the first nucleotide of the cap is a backwards G. I draw it backwards, but my font doesn't do that. And it has the 7-methyl, it's a 7-methyl guanosine. It has the N7 methyl group, like an N7 methyltransferase would add, which is how it got there, the cell's N7 methyltransferase. Then it also has on the next nucleotide, and so the next nucleotide can be whatever it needs to be. That one's going to have the 2'O methyl group, so that's the M2'O. And the N means any nucleotide. And so there you go. And that's a full N7 and 2'O methylated cap structure. And you find those on host cellular RNAs in, higher eukaryotes like us. We consider ourselves high. Yeah. So, yeah, take that, all you lower eukaryotes. Yeah. Too bad, yeast. Okay. Anyway, so first step, the nucleoprotein will bind to that little cap. Great. Nucleoprotein in arenavirus is like a delivery service. It's going to bind the cap, and it's going to take it over and say, here you go. And it's going to hand it off to PA and the PA equivalent and the PB2 equivalent. Yeah. Let's see. Nucleoprotein plus RNA is going to interact with endonuclease, which is the first domain. They sometimes call it domain 1, but it's PA-like. And it uses manganese. That's the Mn2 plus atoms, manganese ions, in order to do its dirty, dirty deeds. Okay, next step. The endonuclease is going to cut off. It's going to pull like 4 or 5 nucleotides past what it wants, trim it off, and it's going to look for nucleotides after 4 or 5 that match the start of the transcription site. So that's how it picks which messenger RNA it wants to decapitate. I imagine hapless, incompetent NPs, nucleoproteins, just bringing RNAs, like, here's one, here's what I found, here's one. And yeah, the L polymerase just saying, no, that one will do because it matches. Yeah. Because as far as we know, N is not smart about how it picks them, but I don't know. Maybe we'll figure something out in a couple of years. Okay, after that, we have got this thing. We have cut the cap off with our endonuclease. We have passed the cap down to the PB2-like C-terminal region of the L protein. And that's going to hold on to it during transcription. And we don't have to add any methyl groups or anything because they're already there. When you steal a fully functional cap, you get a fully functional cap. It's like ripping off a hot rod. Don't rip off hot rods. Stay in school, kids. All right. Thanks. That was your public service message. Okay. Next, the main polymerase domain, which is a normal RNA-dependent RNA polymerase with two kind of shady friends, yeah, on either side of it. We'll then just use this as a primer and synthesize the RNA. Last step in this is also super weird, so why the heck not? Because it's an arenavirus, it can't do anything the normal way. And so it's going to read up to a little stem loop structure because it has, remember, two genes pointing in either direction on each RNA. It has two RNA strands, each with two genes that kind of, one's forward, one's backwards. They sort of face each other. Weird way to do it. They call it ambisense. It's not really plus or minus, but it replicates as if it's a minus sense, and it has a polymerase of a minus sense virus. So it's minus sense. It's just pretending. Yeah. Putting on airs. Anyway, this thing is going to copy and copy. And then in between the genes, there's this stem loop structure, about like so. It's going to copy the stem loop structure. It actually copies it. It doesn't stop before it. And when it gets to the bottom of it, it stops. And that is the 3' end of the RNA. And you're probably saying, oh, wait a minute, where does it get the poly A tail? The answer is it doesn't have one. It doesn't make a poly A tail. Somehow, this stem loop structure binds to the ribosome, presumably, or some ribosomal factor, and I don't know if this is known for certain yet. It probably is. I have probably just missed a paper somewhere out there. But this thing binds to the ribosome in a way that allows the messenger RNA to be translated, just as if it had a poly A tail. So it's a stem loop instead of a poly A tail, which is bonkers. Yeah, it's all kinds of bonkers. But hey, it's viruses. Of course they are. Not going to do anything like a normal, sensible way ever. No, it's not how they work. Okay. Next will be your influenza. And the influenza here, let me move myself over a little bit. There we go. Going to basically start out the same way. Only in this case, the nucleoprotein is not a willing accomplice, as far as we know for influenza. It has to be there, but it's not directly feeding the cap in there, or not directly binding the cap. We're not exactly sure if the nucleoprotein is a cap delivery service or not. It just would make some sense. The PB2 is going to bind to that cap, same old cap, with the two methyl groups, N7 and 2'O, from a cellular RNA. In this case, the virus is actually going to replicate in the nucleus, even though it's an RNA-dependent RNA polymerase, and it doesn't actually use the host enzymes like you would think. So it doesn't really need to be there, but it wants to be there. Why? So it can steal. Yeah. And so this is influenza. It's going to steal that U2 snRNP that's going to form part of the splicing complex at the nuclear pore. And there's enough of these that it'll still work. And the virus has another way to get out. It's just going to ram its way out covered in protein, basically. Yeah. It's a very violent little virus. But yeah, it demolishes the U2. So that's really cool. And it's going to take that to make all of the RNAs, messenger RNAs that the virus needs to make. So it's going to feed this cap, is going to be held by PB2. It's going to be stretched out. We're going to stretch out about 10 or 11 nucleotides away, and PA is going to size it up and cut it off somewhere out there. It's going to be guided by base pairing between the 11th, 12th, and 13th nucleotides, if you like, or 12, 13, and 14, to the RNA that is trying to copy. So it's going to look for binding to the end of that RNA after it's stretched it out. If it doesn't find that, we have no idea. It must just chuck it out because it doesn't seem to use those very well. I don't think they would work very well as a primer, because a primer does have to match its template to some extent, but only for maybe 1 or 2 or 3 nucleotides. Primers are, primers don't have to be as big as you'd think from PCR. Okay, PA uses 2 manganese ions. Again, just like domain 1 of arenavirus uses 2 manganese ions to do its evil business. It's going to cut off 10 or 11 nucleotides, plus a couple that match. Those are then going to be extended like a primer by the PB1 main polymerase. In influenza virus case, it doesn't have that little cool stem loop structure in the middle of its 2 backwards facing genes. And so it just has a small poly-U tract down near the very end of the RNA segment. And so it just gets to that, and it's like a slippery sequence. It just keeps slipping back and forth on this over and over and over again, and putting in more and more A nucleotides. There you go. Since U is a copy of A. Yeah, U templates A. There you go. And that leads to poly-A tail formation. And so you get, in arenavirus case, you get capped, not polyadenylated messenger RNAs. And in influenza virus, you get capped polyadenylated messenger RNAs. Yeah. And neither of them use the host at all, really, to make these RNA species, which is kind of cool. Okay. Where does this leave the other negative strand polymerases, and how they work? Well, yeah, it's a little different. And if you just look at this thing, you may see some features that make you say, hey, wait a minute. I've seen something kind of like this before. So, here we've got a long exit tube. Do you remember coronavirus? It had NSP8, only there were 2 of them instead of 4 of them making the exit tube, but big whoop. Yeah, it's a long alpha helix that's the exit tube for the RNA. So that's still a thing. And then, do you remember the coronavirus polymerase? It had those 2 important domains in there. It had the main polymerase domain with the fingers, thumb, palm, just like normal. And so that's located here. The fingers are in blue up here. The palm is in red. It's flipped around the back there. And the thumb is somewhere in there. There you go. There's the thumb. But in addition to that, remember that the coronavirus polymerase has the extra NIRAN, N-I-R-A-N domain. And the NIRAN's domain job was to attach a cap onto the RNA. Ebola virus and viruses that are similar to Ebola virus cap their own RNAs. And so they have a capping enzyme. And so they call the capping enzyme the PRNT, polyribonucleotide transferase, because it's going to put one cap nucleotide onto a polynucleotide. So it's really a single nucleotide transferase that transfers it onto a polynucleotide. But hey, names are hard, right? Yeah. But it has that function, and it has one more function. So you remember in coronavirus, we had a helicase that could convert GTP into GDP. Whether or not that's the actual method it uses to get GDP, we don't know. But it's there. Maybe it is. I don't know. Then, in this case, there is actually a domain, and it's part of the PRNTase, the polyribonucleotide transferase, and it is a GTPase, so it'll convert GTP into GDP. There you go. So, boom, terminal phosphatase activity, done, sorted. Next activity, guanylyl transferase, right? And so that's the PRNT does that. It just sort of flips that G around, bam, pops it on. Neat. That's the first 2 steps of capping out of the way. What are the next steps of capping? Those are methylation, right? N7 methylation and 2' O methylation. Now, whereas something like a coronavirus specializes, it has separate enzymes for each of these functions. Some of these viruses, like, I believe alpha viruses do it, and definitely Ebola viruses do it. I want to say flaviviruses do it as well, but I would have to look that up. They will go in and they can actually transfer methyl groups to two different places. They can transfer the methyl group to the N7, and they can transfer the methyl group to the 2' O of the next nucleotide. It's a dual function methyltransferase, and that just makes life a lot easier. Why didn't coronavirus think of that? I don't know. No idea. Viral evolution is chaotic. But yeah, so the C-terminal domain is a methyltransferase. Here's our MTAs. This is the Ebola virus L protein genes. You see we've got our actual polymerase up here near the front. Our PRNTAs, that's like our transferase in the middle. Whereas in coronavirus, those were in the opposite order. The NERAN was actually out front, and the main polymerase came after it. But yeah, whatever. They're not all the same. They've slightly rearranged the same parts and come up with the same answer. And then down here we have the methyltransferase, only it is a dual function methyltransferase, which is super cool. So there you go. That's polymerases of negative strand viruses. A little comparative virology to get you through your day, because I know that's the sort of stuff that keeps you going, gets you fired up. All right. Thanks very much. Hope this was useful. I think this will be useful for the test. I'm pretty sure this will be highly useful for the test. All right. See you later.